Well, today we're going to talk about cytotoxic T cells. So there are several learning objectives today. We're going to describe the characteristics of cytotoxic T cells, including the MHC, major histocompatibility restriction, development, and the activation and function of these cells. We're also going to talk about how dendritic cells activate naive cytotoxic T cells and describe the sites where naive T cells and dendritic cells interact. We'll also go through the three signals required for optimal activation of naive T cells. Optimal is a key word here. Describe serial killing, and we'll compare and contrast the three mechanisms used by cytotoxic T cells to kill target cells. As part of that, we will explain the functions of the perforin and granzyme, and we will describe how tumors and viruses can evade cytotoxic T cells. Now, cytotoxic T cells are cytotoxic T lymphocytes. A vast majority of these cells express CD8 co-receptor on the surface. So that's why you see them as called CD8 positive T cells. They're positive for expressing CD8. And these cells are restricted to the MHC class 1 molecules. So those T cell receptors will bind to a peptide in the groove of an MHC class 1 molecule, not class 2. Just remember, in humans, the class 1 molecules are HLA-A, HLA-B, HLA-C, so HLA-ABC. Okay. These cytotoxic T cells develop in the thymus, as all T cells do. They circulate through blood, lymphatics, and secondary lymphoid tissues. Initially, they become activated by dendritic cells and in the secondary lymphoid tissues. So they circulate through, and they become initially activated when they circulate through those secondary lymphoid tissues and recognize the antigen they're specific for. After they become activated, they can now travel to sites of infection to kill infected cells or sites of tumor growth. So they get activated in secondary lymphoid tissues, and once they get activated, they can now migrate into other portions of the body, and so they can go into sites where there's virus infection or cancer cells, and they can directly kill those cells. Natural killer cells, and there are some CD4 positive T helper cells that are also cytotoxic, and we'll talk about the NK cells more later. Now, the routes of antigen processing and presentation by dendritic cells are multiple things that you should be aware of. So dendritic cells can present extracellular bacteria. They get picked up, and they get processed and presented on MHC class 2 cells, MHC class 2 molecules on the surface, where they can be recognized by CD4 positive T cells. They can pick up extracellular bacteria, antigens, etc., and process and present it in class 2 and activate CD4 cells. Now, dendritic cells can also be infected with viruses. And once they do that, those intracellular proteins that are there in the cytoplasm can be processed and presented as MHC class 1 molecules. And in this case, they present antigen to CD8 positive T cells. And there's something called cross-presentation, and this does occur. It's not highly frequent, and it's more with certain viruses and stuff. But they can be picked up by endocytosis, the viruses. So you would typically think extracellular antigens would primarily go into the MHC class 2 pathway. Well, in this case, these viruses can escape out of those vacuoles, get into the cytoplasm, and when that occurs, they then antigens then get expressed by MHC class 1. Okay, so this is a case of an extracellular antigen gets picked up, and the antigen somehow gets into the cytoplasm. So viruses, they have mechanisms to do that. And there are even cases where you can transfer from one dendritic cell to another dendritic cell directly. And those could be presented by the MHC class 1 as well. So dendritic cells are good at presenting antigen through class 2 or class 1. They're very potent antigen presenting cells. And so what typically happens is that antigen gets picked up in some location, some tissue or whatever. They get picked up by that antigen or virus or whatever. It gets picked up and carried to a draining lymph node through the lymphatics, as you can see here. And once they get into the lymphatics, that dendritic cell's sitting there waiting, and T cells will migrate through those lymph nodes, and they enter through what we call high endothelial venules, or HEVs, and they will enter into the lymph node, and they will go through and basically interact with every antigen presenting cell in that, or many of the antigen presenting cells in the lymph node. And if they don't recognize antigen, they can leave that lymph node and go to the next one or back into the blood. However, if there's an infection and that T cell is specific for something with that infection, they will pause and interact closely and begin to be activated and proliferate and expand and develop into what we call effector cells. Okay? So again, an antigen-presenting cell, such as a dendritic cell, will pick up the antigen, carry it to the lymph node, where it holds it there while naive T cells wander through there, and if they happen to recognize that, then they will become activated and expand and differentiate to an effector cell, and that effector cell can now leave that lymph node, go into circulation, enter back into, say, the infected tissue or whatever. Now, to activate a CD8 positive T cell, optimal activation involves 3 signals. Okay, optimal activation. They can get activated with 2 signals, but optimal activation requires several signals. So basically, the T cell receptor will interact with the MHC class 1 molecule carrying the peptide that T cell receptor is specific for. When that occurs, the CD8 molecule will interact with the class 1 molecule. And so that's the first signal. Okay. You now have that first signal there. And there will also be cell adhesion molecules that will hold that cytotoxic T cell and the target cell, such as the antigen presenting cell, together. Okay. And so what happens is pathogens will induce expression of co-stimulatory molecules on antigen presenting cells. This will be dendritic cells, macrophages, B cells. And so pathogens will induce that co-stimulatory molecules. And when that occurs, you can get activation. Okay? You can also induce those co-stimulatory molecules through adjuvants that are added to vaccines. And those adjuvants will also stimulate the antigen-presenting cells to express co-stimulatory molecules. So signal 1 is the T cell receptor interacting with MHC along with the peptide it's specific for. The co-stimulatory molecule will be CD28 on the cytotoxic T cell, will recognize B7 expressed on an antigen-presenting cell. And then the signal 3, to make an optimal activation, is you can have cytokines that are produced by antigen-presenting cell or other T cells. T helper cells, for example, will help activate those cells. And interleukin 2 will induce proliferation of activated CD8 positive T cells. So let's go through this in a little more detail. Okay? Again, the first signal is class 1 molecule carrying a peptide that is recognized by the T cell receptor, and the CD8 molecule will now bind to the MHC class 1, and that's your first signal. So, one of the ways a CD8 cell can get activated is if the dendritic cell expresses high levels of B7 on it, or the antigen-presenting cell. And dendritic cells are well known to express very high levels of B7 once they get mature. And so the dendritic cells express high levels of B7, and that could directly activate the CD8 positive T cell because of that strong CD28 B7 interaction. Okay, so CD8 T cells require a stronger co-stimulation than T helper cells. Now, another way that CD8 T cells get activated is the T helper cells. As I say, the Th1 cells that help induce cell-mediated immunity, they can produce cytokines that interact with the antigen-presenting cell and stimulate them to express higher levels of co-stimulatory molecules. So in the cartoon here, you can see a CD8 cell recognizing MHC class 1. There's no real B7 on the surface in this cartoon. However, if you have an effector CD4 T cell, that's one that has been through the first stage of differentiation, has already been activated, they're now able to recognize a molecule expressing MHC class 2, also presented on that same dendritic cell, and it could stimulate directly the dendritic cell to express B7, high levels of B7. Okay? So this is a case where a CD4 T helper cell is helping a CD8 cell become activated indirectly through stimulating the antigen-presenting cell to express B7. The other way that a CD4 cell can help is by producing a cytokine called interleukin 2. Interleukin 2, if produced at high enough levels, and this will be locally, they could be right, like the CD4 cell could be right next to the CD8 cell. Interleukin 2 can directly interact with CD8 cells and help provide that second signal for activation. Okay. Now, these are three different mechanisms that most likely in vivo, they're all combined doing the same thing, doing it. So you probably have cases where CD4 cells are also helping CD8 cells get activated directly through cytokine production and indirectly through stimulating antigen-presenting cells. And so you can, these three mechanisms, probably there's various degrees where they are occurring simultaneously. But the key thing with CD8 cells, you need a stronger signal than CD4 cells, and it makes sense. CD8 cells can directly kill cells, and you want to have a little more control over those than CD4 cells. Now, once activated, so you've got these naive CD8 cells who've become stimulated in recognition, so you have the stimulation of the naive T cells. They recognize this, and then they proliferate, and they produce interleukin-2 once they become activated. And that interleukin-2 could work on other cells, neighboring cells, as well as themselves. So it's self-stimulating. And it stimulates the cells to proliferate and to differentiate. Interleukin-2 used to be called T cell growth factor. So these naive T cells get activated, they proliferate, and they expand, and they differentiate to effector T cells. Now, effector T cells do not need to have the co-stimulatory signal to become activated anymore. And so those effector T cells will wander around the body, and they will find those cells, like the virus-infected target cells. They'll find those cells, recognize those infected cells, and kill them. Okay? So just keep in mind, you got naive T cells that migrate to lymph nodes and other secondary lymphoid tissues, and when they get activated and differentiate to effector T cells, they can now go anywhere in the body, and going back to the source of the virus, and they will target the cells that are necessary, that are already infected, and kill those cells. The other thing to know about cytotoxic T cells is they can kill multiple target cells, serial killing. So in this cartoon, you can see the cytotoxic T cell recognizes a virally infected cell and tells the cell to die, and then it'll go into the next cell that's infected and do the same thing, and to the next cell. So they're not only a one-shot thing, they can kill multiple cells. Now, as I just alluded to, cytotoxic T cells kill cells through inducing them to die, i.e. promoting apoptosis in the target cells. Okay? And there are three different mechanisms that cytotoxic T cells can use to do this. One is they have granules, what we call lytic granules, and those little granules contain perforin and granzyme B, A, and B. And so when they recognize the target cell, they'll release some of these granules, and they will expose the membrane of the target cell to these components. Now, what perforin does, it forms a pore in the target cell, so multiple molecules polymerize and form a hole through the target cell membrane. And what that does is it allows the granzyme A and or B enter the cell through the pore. And these enzymes will interact with certain other proteins that induce apoptosis, and those proteins are called caspases. Okay, so you can do it through the release of lytic granules. Now, another mechanism is via fast ligand, fast interactions between the cytotoxic T cells and the target cell. So fast ligand you find on the T cells, and fast is on the target cell. So these are molecules on the surface. And so when the fast ligand engages fast on the target cell, it tells the cell to go through apoptosis. The other mechanism which cytotoxic T cells can kill target cells is through the production of certain cytokines, particularly the tumor necrosis factor, or TNF family of cytokines. And they got their name, tumor necrosis factor, for their ability to kill cells. And they basically interact with the receptors on the surface of those target cells and also tell them to go through apoptosis. So let's review these. So one mechanism is you have these vesicles that are in the cytoplasm of cytotoxic T cells. They contain perforin and granzyme A or B, and the perforin gets released and forms channels, pores through the membrane. That allows the enzyme to get through into the cytoplasm of the target cell, the granzyme A or B. And so the granzyme will interact with another protein called caspases, one of the caspases, and when it does that, it stimulates the process of apoptosis. And so here's the more detailed signal transduction pathway. The perforin granzyme interacts, and so you can see there's perforin in the membrane. You can see granzyme A and granzyme B going through that pore. They then interact with several things, including caspases, and the caspases stimulate apoptosis. I don't want you to learn this pathway in detail, but you should understand that what perforin and granzyme A and B do. Okay. And FAS and TNF receptor-mediated apoptosis has a similar sort of story. So FAS on the surface of the target cell interacts with FAS ligand on the cytotoxic T cell, and that stimulates a signaling pathway that includes activation of caspases, which then results in apoptosis. The same thing with tumor necrosis factor. Tumor necrosis factor will interact with its receptor, TNFR1, and in doing so, it stimulates a signaling pathway that includes caspases that trigger apoptosis. And again, I don't want you to memorize these signaling pathways. Just understand that interaction with either of these receptors, the FAS or TNF receptor 1, results in activating some sort of caspase, and with that it stimulates apoptosis. Now, another cell that can actually help stimulate killing are Th1 cells. So inactivated Th1 cells can produce interferon gamma and CD40 ligand and help activate macrophages. So we've kind of talked about that already. And it can produce interleukin 2, which induces T cell proliferation, and you get more T cells, effector T cells. But they also can produce FAS ligand on their surface, and they also can secrete TNF beta. And so certain Th1 cells will do that, and they can kill cells, particularly chronically infected macrophages. And in doing so, they release those intracellular bacteria, which can be destroyed by other mechanisms, including healthy macrophages and antibody-mediated type attacks. Now, one thing that some pathogens in tumors do is they try to evade CD8 positive T cells. And they can do this by downregulating MHC class 1 expression. So if you don't have class 1 expression on the surface, they can't present antigen to CD8 positive T cells, so those cytotoxic T cells will not be able to recognize those infected cells anymore, and therefore they're not as very effective in being killed. Some tumors express fast ligand, and so they can actually do the reverse. They can tell the cytotoxic T cell to go through apoptosis. So fast will be expressed also on T cells. And so if the tumor expresses fast ligand, it may interact with the T cell first to tell it to go through apoptosis. You will also have some tumors that produce certain anti-inflammatory cytokines. An example of this is TGF-beta. TGF-beta is now known to downregulate, dampen immune responses, and will, including dampening inflammatory responses as well as activation of T cells. Viruses can also prevent cell surface expression of MHC class 1 as well. And so they actually have mechanisms that interfere with that. And then there are some viruses that become latent within cells. If you remember, herpes virus is an example. They'll infect a cell and they will hide inside that cell and be dormant until the immune response kind of becomes low, but not as effective. And so we'll talk about natural killer cells, which can come in and play a role in helping address some of these strategies as well.